in physics at Cornell University. After postdocs at the University of Karen, the Cape Valley Institute for Theoretical Physics, and Stanford University, he joined the Department of Human Genetics at McGill and the Quebec Innovation Center in 2013. Simon's interested in learning about biology and evolution through the creative analysis of high throughput biological data. His group develops mathematical and statistical models that take advantage of diverse data sources to refine our understanding of fundamental parameters of human history and biology. Today he's going to talk, well, I'll let him say what he's going to talk about, but his recent research has focused on how the history of diverse human populations affected patterns of genetic diversity and disease. And his group has projects focusing on anthropology and history, technology development, biology and medicine. He's also involved in the Carthagène project, so he's everywhere in Montreal genetics. We're really excited to hear his talk today, and so I'll let him take it away.
Thanks, Morgan. Thanks, everyone. It's a great pleasure to be here, and I'm seeing that I have both the French and the English title here, and I think the French title is a bit more revealing than the English one.
We don't see your slides, though. Oh, sorry, Simon. Yes. We don't see your slides.
You don't see my slides? Oh, no.
We should have kept them.
I need to restart that. Okay, no problem. Hold on. Okay, great. Yes. So I'll be talking today about early human history, as I mentioned. And as the image in the background is suggesting, we'll be trying to pick apart very tangled roots of our species. The more we look into human history, the more complications we find, and it seems to be kind of a fundamental property of our history. I will start with my acknowledgement slides because I tend to get sidetracked and not get to the end of my slides. Most of the work that I'm going to be talking about today was done by Aaron Ragsdale, who's now faculty at UW-Madison, and actually, so this is a much older photo of him, but if you look through the participant list, I just noticed that he is here with us today with a slightly more recent photo. So if you want to see that, you can look through the participants. Of course, we also had help from other collaborators. This project was kind of started by another postdoc, Julien Juganus, and I'll introduce some other collaborators along the way. But of course, I didn't do most of this work, and I certainly didn't contribute all the samples either, so thanks to the participants as well. Okay, so the first goal that we're going to be trying to reach in this talk is to learn about early human demography, right? So this is where this project all started. So this is a slide with a figure from Brenna Henn, where she outlined what was known in 2012 of the broadest strokes migrations through which modern humans who appeared in Africa, sometime maybe between 100 and 200,000 years ago, and expanded throughout the world, right? And if you look at this, even though modern humans appeared in Africa, again, at the time I just said, most of the arrows we have here are outside Africa. We tend to know more about human migrations outside Africa, partly because populations outside of Africa have been better studied, and partly because it's easier, in a sense, to model histories that are more recent, because there's less stuff that has happened. And by contrast, we don't have as much information about what happened in Africa, even though there's way more history happening there, just in terms of duration. And so this was our goal, kind of running into this project. Now, of course, this is not quite true, right? I'm doing some historical revisionism here, in the sense that this project here started really with us doing some mathematical exploration, trying to solve a completely different problem in medical genetics that had nothing to do with history in Africa. And in doing that, we kind of randomly stumbled upon these interesting mathematics and these mathematical tools that we eventually figured out, were going to be helpful to learn about early history. But the story is a lot better if I pretend that actually this is what we were trying to do in the first place, and if we knew what we were doing all along. And senior scientists in the room are all familiar with the fact that often this is how we present our talks. But I'm being explicit about here for students who might be confused about like how did we know to develop that math, like starting from this problem, and it doesn't make sense. It just happened in a slightly different way. Okay. And so I'll pretend we started with this problem of learning about early human history. And there's been a lot of discussion in the genetics and anthropological world about how we should even try to model populations. And so this is a slide from a couple of papers by Lunes Chiki and collaborators, where they considered the present at the top here. And imagine we have 3 different populations in the present. And what's going back here, what these little tubes are showing is our imagined model history of these populations going back in time. So a tube corresponds to a kind of cohesive population in some sense. And the point they were making in these reviews, which is a very good point, is that most geneticists tend to think about history of populations in models that look like B here, where we have present day populations. And as we walk back in time, these merge together, into ancestral populations. And as you go further back in time, everything becomes very simple. And we have like one ancestral population at some point in the past. And we can ask questions about like, when did this ancestral population exist and where was it located? And this is convenient for geneticists because it's a reasonably simple model that we can do math with, and we can fit data to. But the argument that they're making is, well, it's much more likely that if things are complicated today and there's geographical structure, everywhere we look, it's fairly likely that this was true as well, arbitrarily far back in time. And there's nothing to say against that. I completely agree. They also go on to look at different kinds of archaeological artifacts that support this hypothesis. So they look at different stone tools or ornaments, for example, that have been identified in different parts of Africa. And I'm not an expert in interpreting those, but the big picture here is that you find similar kinds of tools and artifacts that pop up and disappear in different parts of Africa. And so if you want to have a model with a single ancestral population that is kind of unified, it has to do a lot of running around Africa over hundreds of thousands of years and bring its culture with it all along. And in a sense, these patterns of artifacts are easier to explain if you imagine that you had all kinds of populations, appearing and disappearing and merging and splitting and so on. So that it's complicated is the argument here. We have a different line of evidence from the paleoanthropological record, in the forms of bones that we find. And so this is a figure from a 2017 paper that described different reasonably complete crania that have been identified in Africa. And I'll point out the legend here, where the orange dots represent crania that kind of look like modern humans, Homo sapiens, and the date, right, where they've estimated to have lived. So 300,000 years ago in Morocco, 195,000 years ago in Ethiopia, and 120,000 years ago in Tanzania, right? So, and then we have other crania that look morphologically a little bit different, that look more, more archaic from our perspective, which is going to mean, from my perspective here, just more different from Homo sapiens. So we have them as well in different parts of Africa. And similarly, so these have been classified by anthropologists into different categories. So maybe there's one here that might correspond to maybe a species called Homo naledi, and there's a couple that might look like another maybe species that would be called Homo heidelbergensis or other species. And it's very debated what's going on there. Are these different species? Are there descendants of each other, or how are they related? We don't really know. Again, the point for this talk is it's complicated. If we want to have a model with just one ancestral population that is kind of reasonably small, it would have to have kind of, if we believe these results, the population would have had to be established in Morocco 300,000 years ago, and then have moved to Ethiopia, potentially 200,000 years ago, and then to Tanzania. It's getting a little bit complicated to accommodate all of that. It's not impossible, though. Now, other observations, other lines of evidence why things would be complicated are from models of the climate. So we have pretty good models of how hot or cold things were, or how wet or dry, based on all kinds of things I'm not an expert on either. But based on climate models, this particular paper has established, so they separated Africa here in 4 main regions: North Africa, East Africa, tropical Africa, and Southern Africa. And what you're seeing here is, over time, what were their best estimates of whether the conditions were fairly dry or fairly humid, with the idea that more humid conditions are more amenable to human populations. And the point is that things change over time, over the course of tens of thousands of years. And so if you imagine that population sizes or culture or history depends on these climate change events, it's natural to think that populations didn't stay put for hundreds of thousands of years. Okay, so there's lots of lines of evidence supporting the idea that things are complicated. So main conclusion is it's unlikely that we had a single ancestral population, and the emphasis of these papers was that, well, we shouldn't try to fit these single expansion models. We should try to fit models like these ones, right, that have a lot of populations splitting and merging. So this is something I completely agree with. And actually, this is something that maybe people here, there's lots of people working in complex systems, I think, in this group, might be able to give us ideas, because I think as a field in population genetics, I feel a bit stuck here, because as much as I believe that these models are much more realistic, I don't know how to learn these kinds of models. I don't know how to fit them to data. Whereas this model here, which is much more simple and probably more wrong, I know how to fit to data. And the question is, what's the most useful? Is it most useful to fit a wrong model to data, or to try to fit a more realistic model that might have too many parameters for you to know what to do with? And just to really belabor this point, this is a map of the world from 1507, which we can recognize pretty well. It's a pretty good map for the time. And you can see, we can see Africa here, which is the area we're interested in. But from a map making perspective, we can look here at what's happening in North America, where we have something going on that's pretty, recognizable on the east coast of North America, of the Americas. But there's all kinds of weird things going on here. It's not quite accurate. So when they didn't know what to do on the West Coast, they just kind of, I will say, oversimplified in a sense. So the map is lacking complexity here. Okay, so that's one option. And the other option you can do, figuratively, is, well, if you don't know what's going on, you can just invent stuff, right? And this is kind of a map, a classical medieval map where people just added all kinds of crazy animals, which might have been partly decorative as well. I don't want to throw shade at the author of that map. But the point is, if you allow too much stuff in your model like this, in the end, you don't know whether what you're learning is going to be something informative about history, or is it just going to be monsters that you've invented accidentally by fitting parameters? It's a hard problem, and I don't know how to solve it, but we can do things about it. So we can try to learn things. So So this is the philosophical part of the talk. So where is the field at? Like, what are the kinds of models that people have fit to data using human genetics? And well, for many years, people have been fitting models like these ones, where you have these pipes that represent individual populations. And I apologize, I've been changing the direction of time in a lot of these, even within this slide, so it's a bit confusing. On this particular figure here, times, this is the past and this is the present. And on that figure, this is the present here, and this is the past here. Apologies for this. But so on both of these, each pipe represents a population. And so we have these present day populations for whom we have sequence data. We're going to have a number of parameters representing population sizes, and times at which events happen, and split times, and migration rates, and growth rates. And we're going to learn we're going to put a lot of parameters in these models, and we're going to try to have these models fit the data. So this 2009 model by Ryan and Gutenkunst was like a model that has been used a lot to try to just understand the broadest strokes of human variation and the consequences of the out of Africa event, meaning the observation that populations who currently live outside of Africa and who have been out of Africa for over 10,000 years have reduced genetic diversity as a consequence of having experienced a genetic bottleneck at this point. Okay, so that's one kind of model that's still very, very simple. Over time, people have added pipes to these models and making them more involved and more complicated. And one relevant model here is shown on the right, which is just a generalization of this model. We still have a single population in Africa, but now we have a lot of complications about what's happening outside of Africa. In particular, as many of you have heard, people have found pretty good evidence at this point that there's been admixture or gene flow between Neanderthal and modern humans outside of Africa, and also another species called the Denisovan. And so we've now starting to have a model that is pretty complex and reticulated outside of Africa, and it describes a lot of the patterns of genetic diversity that we're observing there pretty well. But now this model, of course, is still like treating the African populations as a single uniform group going back, I don't know exactly what the time is here, but at least 70,000 years or something like that, which is not particularly realistic. Okay, so now we've, of course, people have also looked at variation within Africa. And so if we try to build a model like this with pipes and arrows and so on, it could look like the one we have on the next slide here, right? So we can imagine, okay, so we have this Neanderthal branch admixing into Europeans who are shown here. And maybe at the broadest strokes within Africa, we can imagine considering populations living in South Africa, in West Africa, and in East Africa. And again, this is a vast oversimplification here. And then people have built models like these ones and have asked questions like, okay, so if we do this and we look at different populations within Africa, how closely related are they, right? How much genetic variation is there? And so that's something you can empirically measure. You can count genetic differences between individuals. But also people try to learn, okay, how far back in time would these populations have potentially split according to our models to predict this amount of genetic differentiation? And so there's lots of papers that try to do that. And in this review paper here, collaborator Brenna Henn, just tally these estimates of how far back these splits might have occurred, with the idea that if we have a model of what we call a recent expansion model, where all populations within Africa have a recent-ish common ancestor, we should kind of consistently find that time, in our estimates of divergence. Whenever you consider, say, South African populations with any other populations, the split time should be kind of consistent. And message of this table here is that these estimates are kind of all over the place. With estimates, even when you consider the same data and the same populations, like the San in South Africa and the Yoruba in West Africa, we have estimates going from 90,000 years to maybe 140,000 years, and then you can even push it with slightly different populations to 200, 300,000 years. And so there's all kinds of things that might explain why these estimates are so different. People are using different statistical methods, they're using slightly different data, and they're using slightly different values of parameters in their estimates, like what's the mutation rate, for example. But one very natural explanation for why, if you take a model and when you poke at it, it gives you very different estimates, it might be because it's just not the correct model, right? And maybe this model of a recent expansion is not a very good model for capturing diversity within Africa. And so the idea here is we'll be trying to get together the genetic information and some information from archaeological and paleontological data. And so to do this, we've been working, well, of course, with Aaron, also with collaborators Brenna Henn and Tim Weaver from UC Davis, both from the anthropology department. So Brenna has training in anthropology, but is also a very competent geneticist. And Tim Weaver is really an expert in evolution and paleontology. So he knows about bones and things. And so we've been trying to work together to define, okay, what does the anthropological record tell us? And how can we go and test these different models that are consistent with that record using genetic data? And so what they tried to do is Brenna and Tim went and read this literature and tried to come up with models that people had proposed. And I apologize for the noise, but the daycare is closed today, but baby is being taken care of in case you're worrying. Okay, so some possible models that have been proposed. Well, one of them is this recent expansion model where we had a single human population maybe living 120,000 years ago or something like that. There's also the idea that there was this kind of expansion, but with this expansion, population expanding, say, from South Africa, as they were expanding through different regions of Africa, met with other populations that already lived there, that were diverged for a longer amount of time. And so there was some amount of mixing going on. So that's a very plausible model. And that could explain some amount of cultural continuity that seems to predate the 120,000 years ago and some of this cultural variation we're observing. And then there's a question of how much cultural and genetic contribution would have co-occurred. A model that has been discussed a lot is the model of archaic admixture. So I showed you some of these, the slide on crania, and there was evidence of humans, hominins, right? Essentially humans that are fairly diverged from modern humans, that look fairly different, that lived in Africa at that time. And so because they're morphologically different, we can kind of infer that they probably have split a very long time ago. But maybe if they coexisted in a given territory because we have evidence that, say, Neanderthal admixed with modern humans outside of Africa, despite being deeply diverged, then maybe the same thing happened in Africa. And there's a fair amount of genetic evidence supporting that, or something like that. And then the other one would be a multi-regional model where there were populations, modern human populations living in Africa, going back hundreds of thousands of years, mixing over time, but essentially, there is continuity of populations and maybe cultures in different parts of Africa. And so these are the different models that people have discussed in the anthropological literature. And so now what we've done in this slide is translate these models into these little pipes and arrows models that we know how to test using genetic data. And so that's going to be the topic of this talk. We'd like to see how well these different models are able to represent what we're seeing. So The challenge, though, is that these are pretty complex models and they have a very large number of parameters. And so we're going to have to try out a lot of models. And so we're going to need to find out a way to quickly test these models. And this is where the math will come and help us. There's a philosophical question, or I don't know, it's philosophical. It's a data science question of how we explore these different models and when we think we've explored models enough when we have these very complex systems. And then I'll conclude, of course, with what we think we might have learned. So testing models. So the framework here conceptually is pretty simple. So we have a model with these pipes and arrows. And these arrows represent migrations. The size of these pipes represents population size. Excuse me. We're not going to have natural selection in our model. We're going to assume that we're going to look at parts of the genome for which we don't have evidence that there's a whole lot of selection going on. But everything else we have in the model, we have dozens of parameters. So that's the model. On the other hand, we have genetic data from humans from around the world. So we recruited people, or we use data that other people have recruited. We sequence their DNA, and now we have a whole bunch of sequence data. And so we have these observations. In principle, from this model, we can generate predictions about what we should be seeing. And then we're going to compare the two and trying to find parameters that agree with our observations. It's very conceptually simple. So for people who are not in population genetics, the model itself is actually pretty straightforward. It's called the Wright-Fischer model, in which we have a parental population, and each parent in humans, we're diploid, we have 2 copies of the genome. And when we generate an offspring, each parent generates a gamete, and the gamete is generated by copying from their own 2 copies of the genome, using a simple, mathematical, like a Poisson process to generate the gametes. And then each offspring gets 2 parents, and essentially what happens in this model is that each offspring picks 2 parents at random from the population at large. And when you do this, it's very convenient because each parent can be selected, never or once or twice. And so the stochasticity accounts for the fluctuations in the allele frequencies over time in population. So it's a very powerful model to test fairly general scenarios of evolution. So that's our model. So here's all the parts. If I specify all these parts, and this is actually a slide from my, I think the second lecture in my intro PubGen course, and you can specify all these things and you can go on and simulate data according to this model. It's pretty straightforward conceptually. So this is great. We have a model, we have parameters, we can do simulations and try to see if the simulation matches the data. There's tons of really amazing recent progress on simulating genomes. If you are interested in this but haven't been following this literature, there's a lot of really cool simulators out there. Unfortunately, it's not fast enough for us to run these simulations because we still have to simulate, thousands of genomes or potentially millions of genomes if we want to do a full simulation of the entire population. And it's just not fast enough yet to do this kind of inference. So this is where the math is going to come in. And so there's one other intro population genetics equation that I'm going to walk you through because I think it gives a good feeling of what we're trying to do here. And that's an equation for the probability that if you take 2 copies of the genome from the population, right? So we're diploid, so we each carry 2 copies of the genome. But imagine you take one individual in the population and 1 copy of their genome, and another individual and 1 copy of that genome, and you compare the 2 at 1 position in the genome, and you ask how likely are these 2 alleles different, right? How likely are these 2 individuals to be different at this given position? And there's an equation for that that's completely magical, and it's very elegant. So to compute this, what you can say is, I'm going to condition on whether their parents are the same or are different. So these two copies of the genome, either they could have been inherited at the last generation from the same individual, which is illustrated here, right? So we have these two copies at time 1 of the genome. Maybe if you go back one generation, these have the same parents, right? And that's very unlikely. It happens if you have 2n haploid copies of the genome, it happens with probability 1 over 2n. So it's quite unlikely, but if that happens and there's no new mutation, these two individuals are going to carry the same allele because they've inherited it, they've copied it from the same parental allele. And so in that case, conditioning on them having the same parent, pi, the number of differences, is 0. Okay, so that's the kind of boring case. Now, what happens if the two copies of the genome picked two different parents? And now we're going to ask, well, how likely are these two individuals now to be different if they pick different parents? Well, the two parents were randomly picked from the population, and so their probability of being different is just pi at time t-1, the probability that two alleles drawn at random at time t-1 are different. Okay, so this is, you kind of have to wrap your head around this, but it gives you this little recursion there that the probability that the offspring are different, excuse me, here at time t is the probability that they draw different parents, 1 minus 1 over 2n, times the probability that their parents, their different parents, are different from each other, which is pi at times t-1. Okay, so this is a completely magical equation, right? You can have a very, very large population. I mean, you have 2n individuals, but you can write this recursion only taking 2 individuals at a time, and you don't have to consider all these other 2n minus 2 haplotypes at all in generating predictions about genetic diversity. So one of the classical results in population genetics happens if you say, okay, what happens now if you have mutation happening as well, right? So because I haven't accounted for the possibility that a new mutation comes in. Well, mutation just gives us another thing to try to keep track of, right? It's possible, for example, for these two individuals to carry a different allele. If they both pick two different parents, they both picked a different parent, the parents had the same allele, but there was a mutation occurring here, creating a difference now between the alleles carried by the offspring. So it's purely bookkeeping here. But with this bookkeeping exercise, we now have a recursion for pairwise differences at time t, pairwise differences at time t-1, and then the mutation rate here as well. So we have population size, mutation rate, and so this classical PopGen result will occur if we say, okay, what happens if I have a population that's very boring, whose size is constant over time and is evolving for a very long time, such that things don't change anymore. So if we say, well, in that case, we can assume that the pairwise differences don't change anymore. We set pi of t equals pi of t-1 in this equation, and we can solve. Then we can say, the number of pairwise differences in a constant size population is 4 times n times the mutation rate. Again, this is equation 2 or 3 in a lot of population genetics textbook. So if you have more mutations at each generation, you're going to have more differences. This is intuitive. But also, if you have a larger population, you're going to have more genetic differences across individuals, which is a little bit less obvious. But okay, so this is a very, very classical result. But now what we're interested in doing is considering the fact that that human populations are not a boring constant size population. There's a lot of dynamic stuff going on. And so we were going to want to use these equations, these recursions, but instead of solving them at equilibrium, we're just going to say, well, let's allow for the population size to change over time, for example. And so here, if I have an example where the population is evolving from here with size 10,000 over time, and then it has a bottleneck, I don't know, about 75,000 years later, and then it recovers and has another bottleneck and so on. You can put an arbitrary population size over time, responding to changes in the climate, for example. So these two bottlenecks in this case, we can just solve this recursion very rapidly. And what we're going to see is that in this population, the number of pairwise differences is going to decrease during the bottlenecks, and then it's going to recover, and then it's going to decrease again in the second bottleneck, and so on. So we can have very, very fast predictions about what's going to happen. So we don't need to simulate millions of genomes. It's mathematical magic. So this is great, and this is a beautiful equation. The problem, though, is that after all this work, we have in the present, so this is the present, at the end of the simulation, we have one number, right, which is the number of pairwise differences. And we're going to try to be fitting a parameter, a model, which might have, 15, 20 parameters. And so we don't have enough information to fit a model. So it's a cool equation. I mean, it's been known since like 1940 or something, but it's not enough for us to get going. So where do we get more information? The first thing we can notice is that, well, we're going to have more than one isolated population, right? So we're going to have a model with multiple types. And so we can say, well, what's the number of differences between individuals within a population versus the number of differences between individuals across population, right? And we can expect that there's going to be more differences across populations. And maybe this is going to tell us something about migration rates and split times and so on. So we can do the same kind of game, same kind of bookkeeping. So if we have, say, three populations here, we're going to have 6 pairwise differences, right? We're going to have pairwise differences between individuals within the population. There's 3 of them. And then we're going to have pairwise difference between individuals across populations, and there's 3 of them. And then if you do this bookkeeping exercise of asking, how is it possible for 2 individuals to carry different alleles? You can still do this in this case with multiple populations. You just have to keep track of things like, well, maybe these 2 individuals from different populations had 2 parents, but one of the parents was a migrant here. So the parent of this individual was drawn from this population, and now these 2 parents carry different alleles. So we still can write these recursions in closed form. There's the mathematical reason why we can do that, but I don't have the time really to discuss that today. But now we have suddenly 6 observable statistics for 3 populations. And so if I take the model, for example, of Gutenkunst that I showed you earlier for the out of Africa event, we have all these parameters, and we can generate predictions over time. And this is what we're seeing here with the time in generations. And we're looking at, within this model, actually, I'm not sure if it's the exact same model, but within a similar model, we're seeing the pairwise differences over time. And when there's the out of Africa event, we see this reduction of diversity observed in out of Africa populations and so on. And again, we can do this in no time compared to simulating millions of individuals. And we can go and compare different values of these parameters to data and try to fit these models. So this is great, but we still have only 6 parameters. And we want to learn 6 observables, and we want to learn dozens of parameters. And so it's not quite enough. So what will we do? So there's 2 options. One option that we tried is to say, well, instead of looking at pairs of individuals and pairwise differences, we can look at more complicated statistics involving larger samples. We can say, like, let's take 100 individuals and look at the distribution of allele frequencies within this sample. And we've tried to do that. And it's interesting, but it doesn't scale. It's not computationally performant enough for our purposes here, so I won't talk about that. The other option, which is going to be the focus here, is considering more than one genetic variant at a time, right? So far, what I was asking is I was considering a single variant or at a time, and I was asking how likely is it that 2 individuals are different at one position. And so each individual had only 2 possible alleles they could carry at each position. Now what we're going to do is we're going to say, consider 2 genetic variants at a time. So now we might have 4 different combinations of alleles. We might have individuals carrying the ancestral on both loci, the ancestral at one locus and the derived at the second locus, and conversely, and so on. So we have 4 possible haplotypes for each individual. And all of a sudden, we have a lot more combinatorial possibilities. And so we're going to see that we're going to get a lot more information about history. Okay, so we're going to have to do some bookkeeping again, but instead of doing this this bookkeeping for a single 2 individuals, 1 allele, we're going to have to look at bookkeeping for, say, 2 individuals and then haplotypes here. So how likely is it that my 2 individuals carry, for example, the derived allele here at both loci? Well, one way to do that is that they both share the same parent. It happens with some fixed probability. And similarly, the additional biological effect we're going to have to consider here is the effect of recombination, right? The fact that the parents don't always transmit the 2 alleles together. They might have copied from both their ancestral haplotypes to generate a gamete to pass on to the next generation. And so we're going to have to add a term in our bookkeeping that says, well, maybe the offspring received a haplotype that's been created through recombination. Again, it is a work of bookkeeping, but what is magical is that you do all this work and then you find that you get equations that close, that don't require you to simulate the entire populations. And in particular, if you do this for 2 individuals here, you're going to find a recursion for a statistic called D, which is defined in this way. And I'm not going to go in too much detail because I'm really, really running out of time. It's the number of times you see the ancestral alleles together on the same haplotype minus the times when you see the ancestral alleles on the different haplotypes in a sample of size 2. There is a recursion for that, and it's been discovered in 1966. If you add, a bit more individuals in your recursion and you do more bookkeeping, you get another recursion here for d squared, which d squared, for anybody who's run a GWAS, this is the measure of LD that people typically look at between the 2 loci, how much linkage equilibrium there is. Often in GWAS, people normalize this by the variance, and they look at r squared instead, but it's really the same information. Okay, so we have now a recursion that measures linkage equilibrium in an arbitrary kind of demographic model for a single population. And so people have looked a little bit at the effect of linkage, at whether linkage equilibrium could be informative about human history in Africa, in particular in this paper from 2011. But because since until recently we didn't have a good model to compute this linkage equilibrium rapidly, people couldn't really compare this to concrete models, and they had to make these kind of general, here's a trend, and try to tell a story around that. Okay. So what happened here, I'm going to skip this slide because I don't have time. What happened and what Aaron did here is to do this bookkeeping exercise and realize that you could compute these 2 locus statistics in a multi-population model with splits and mergers and migration and recombination and mutation. And so we have this family of statistics we can compute in essentially no time and compare those to observation from genetic data. And so this was this kind of mathematical observation that, okay, these equations magically come up, we can solve them, we can generate predictions for them. So now let's look at data. In this first, when we first discovered these equations, Aaron looked at something like the Gutenkunst model for out of Africa, right? With 1 Asian population, sorry for the labels here. This is Chinese from Beijing. This is a European ancestry population from Utah. And this is a Yoruba population here. And so we were able to generate predictions for all these statistics that came through the math. and compare those to observations in the data. And what we're seeing here is that for well-known statistic like the linkage is equilibrium, d squared, the model did a really great job at predicting the data. The dots are the data. There's error bars on there. They're very tiny, and the solid lines are model prediction. We looked at other statistics like the correlation and linkage is equilibrium between population, which is relevant when you try to transport GWAS risk scores across populations. And this also the models do very well. What was very interesting is that there was this other statistic that just came through the math. We didn't have any intuition about it. It just like when we did the math, we were kind of forced to compute this quantity. And that quantity really didn't work, didn't have a good agreement between the model predictions and the observations, especially within Africa. So again, you have error bars here. So it's both very, very significant departure. And it's also a large, it's like a 10-fold difference or maybe 8-fold difference between models and observation for this particular statistic if we didn't know what it meant. I'm, again, very short on time. If I make a very quick explanation of what this statistic is measuring, we scratched our heads a lot. So it's the D, the measure of linkage is equilibrium, multiply by 1 minus twice the frequency at the left locus times 1 minus twice the frequency at the right locus. And so it's something like LD weighted by how rare a variant is. These terms are 0 for variance at frequency 0.5. It's maximum for rare variants. So it's in a measure of LD between rare variants. After scratching our heads again a lot, we thought, oh yeah, so if we have this situation with an archaic admixture scenario where a very diverse population accumulated a lot of mutation and suddenly admixed into, say, modern humans, now modern individuals are going to carry these haplotypes, these stretches of their DNA that carry a lot of rare variants, and that would really increase that statistic. And so maybe that's what's going on. That's what's missing in this model. Maybe we should add a branch to this model of an archaic human population in Africa that contributed to African populations, both before and after the Out of Africa event. And if you do that and you add a couple of parameters, you magically explain these patterns rather well. So we're very happy about this. And so if we went back to these slides, and now we want to say, okay, there's an archaic branch here. So who is it? Is it Homo naledi, maybe, or is it Homo heidelbergensis? Like, who is the modern humans that might correspond to this archaic branch? And a lot of other people tried to, who also suggested maybe there was archaic admixture in Africa. We were not the first people to propose that, but people were trying to think about, okay, who could that be? Who could this archaic branch be? Because it's very relevant for early human evolution. Okay, now I've built a slightly more complex model here, right? In the sense that I've added one branch to this model, to this Out of Africa model, and it explains the data better. So if I go back to my initial question of going between these two models, I took this very simple expansion model here, and I added one branch here with admixture. So I'm still kind of far from here. So how can we do this? And again, so I'm going back to this slide here that I had presented earlier, where, okay, so let's look at all these different models that people have proposed and test them, right? Not just this archaic admixture model I just presented, but let's try these other models just in case they are also, they also describe the data very well. Okay, I'm just going to kind of jump straight to the results. There's lots of material challenges here in terms of coding these different models well, making sure that your inference in large number of parameters is robust, and there's no bugs, that things are reproducible, etc. But I'm not going to have the time to go into very much detail here. I'm just going to show. So what we did is that we tried a bunch of models. We have the best fit model, which actually is the one on the right here. But what we did is, instead of just focusing on the best fit model, we tried to look at the collections of models that fit the data reasonably well, and look at what was consistent and inconsistent across them. And so if I show you here, so this is the best fit model. We have populations that I haven't really introduced to you yet. So this is, I went too fast in the previous slides. I'm going to go back to this slide here. So we're going to be looking at populations, the Nama, which is the populations from South Africa, the Gumus, the Amhara, and the Oromo, which are populations from Eastern Africa. We're going to include British and the Neanderthal populations just to anchor, to have a time here for the split. And we also have the Mende population, which has recent ancestry in West Africa. Okay, so these are the populations back here. So we have a South African populations, West Africa here, Gumus, the Gumus and the Amhara and the Oromo here. We have our pipes. We have a whole bunch of migrations between them. And what we find is across our models, we're doing a pretty consistent job at predicting what happened in the last 75,000 years. Like if you take a cut here, right, these two models below that are pretty consistent. You can see there's some differences in how these migrations are represented into the Mende here, excuse me. Here we have a single event of admixture, whereas here you have continuous migration. So this is a consequence of us parameterizing our models in different ways and assessing robustness, but this is fairly consistent. If you look at the more distant past, things are a bit more different. If you try to line things up here, it's a bit harder. What is very consistent across these models is that we need multiple stems. We need multiple ancestral populations to capture the patterns of genetic differentiation that we're observing. So this is consistent and with very high statistical confidence. But what we're seeing in all these models that fit the data fairly well is that we have migration. The models always want to add migrations between these stems and keep them not very diverged from each other. So these will be populations that have ongoing gene flow, over long periods of time. So if we take this model and try to translate this back into anthropological language, what we can say with a high degree of confidence, I think, is that that model of a recent expansion is not supported by the data. There's strong evidence for deep population structure. I would also argue probably that it doesn't really look like an African multi-regional model, in the sense that if we trace back from the present day population, there's a lot of mixing that happens in all these models, such that we can't trace a single population back more than 100,000 years or so. So we don't have evidence for, say, a single population living in South Africa in relative isolation for more than 100,000 years, for example. I had anthropologists tell me that I'm interpreting this African multi-regional model not completely right, so people can push back a bit on that. But now, which model between these two would it be? Like, could this still be? Could these two stems still correspond to something like modern humans and archaic humans? Could this be Homo heidelbergensis? Well, one thing we can do now is in our model say how differentiated these different stems are. Do they look very distinct from each other, like human and Neanderthal, or do they look more like differentiated like modern populations? And so I'm showing you one of these models here. But what we're seeing, so here is the time is going in yet a different direction. This is the present, this is the past. The y-axis here represents the amount of genetic differentiation between pairs of populations in our models. The dotted lines represent the amount of genetic differentiation between Neanderthal and modern human populations. So this can be an archaic human kind of example, a very differentiated population. And the dotted lines, the dashed lines here represent the amount of differentiation between the stems here in our model. And what we're seeing consistently is that these stems are not as, they're not at least not a lot more different than the amount of difference you observe between contemporary populations. All right. So personally, conceptually, what I'm seeing this. What I believe more is we probably don't have something like an archaic model, but more something like a structured population going back, at least 600,000 years or something like that. We don't know that there were exactly two populations. There's too many parameters to fit, but there is structure, there's stuff going on, probably between not two differentiated populations going back 600,000 years. So we did lots of validation of these models using different statistics. I'm not going to talk about that. If we go back to this figure here, and we try to think, okay, so when we look at these different stems that we've been fitting in this model and other people have been looking at, who might correspond to these different stems? And my best guess at this moment is that it's probably not, these Homo heidelbergensis or Homo naledi kind of crania, and it's more likely to be maybe these modern human-looking individuals. Although at the moment, from the genetic information, we have no idea of knowing where these different stem populations might have originated. And it's possible that they both originated from a similar part of Africa. So we don't know that, but this is a speculation at this point. So what can we kind of reasonably strongly conclude? There is structure in the human lineage going back at least about 600,000 years ago. So I don't think that models with modern humans appearing as a single population 120,000 years ago is tenable. These different stems that we have infer in our model, they contributed differentially to different present-day population. So it's not like there was one stem that's in South Africa and one stem that's in East Africa and they're consistent. But there's lots of mixing and lots of reshuffling that seems to be happening, and which we don't exactly know what to do with. There's evidence for ongoing gene flow between these populations. And here I'm just repeating what I just said, and we're low on time, so I'll just stop here. So thanks, everyone. That's all I have to say.
Thanks, Dimal. I mean, obviously you're talking about many, many years. There's a lot of things to touch upon, so it's fine to be to take that much time. We are at time, but I think that it would be nice if you can stay, if we can ask a few questions, if that's okay with you, maybe for 5 minutes.
I'm fine with it. Yep.
Great. I'll start with a question, which is, I don't know if it's, so how were you assessing best fit? Because I think that this is a problem, not just in population genetics, but it's something that we come across in even fitting, deterministic models. Because, for example, in fitting pharmaceutical models to understand pharmacokinetics, a lot of it is looking at best fit. And I really liked how you tried to integrate, not like domain knowledge and domain expertise to try and understand what is actually likely, even though mathematically we might have best fit. So maybe you could talk a bit about that.
Yeah, I mean, this is a really important point, and this is one where I wish I had a more satisfying answer to give. But so what we do is that we do have a reasonably principled way of computing likelihoods, right, in this model. And so, I mean, what we're doing is maximum likelihood fitting. And then when we're doing, model selection, for example, we can use the familiar machinery of, nested models and information criteria and these kinds of things. A challenge we have here is that when we compare the likelihood of models, there's so much data that we always kind of favor the more complex model. And this is the problem where, if we add a parameter, it's always going to be supported but we can add, say, a demographic parameter that's going to improve the fit to the data. But it might just be, for example, because it's capturing some idiosyncrasy of the data, for example. So we don't know if we can interpret these parameters. And so I mean, one big challenge we have is I don't know if a statistical way of addressing this problem that our data is very informative and our model is very complex. And so when we formally test uncertainty in the parameters, these uncertainties are very small because these data are very informative. And most of the uncertainty comes from model misspecification, essentially. So we have our model is just too simple, and it's not capturing some important factor we didn't think of. And so and if we try different models, then sometimes we'll just get results that are very different. And I don't know if a good statistical tool to do this, aside from doing like a full Bayesian analysis over the space of all possible models, but that's way too big for us to attempt at the moment. So, so again, practically the way we've done this is by having this bag of model approach where we say, okay, well, we don't, so most population geneticists will say, well, we fit one model, and then we, here's our result and here's our uncertainties. And what we try to do here is to say, let's fit 4 different or 10 different kinds of models and parameterize them differently. And that's our stress test, and try to figure out what's consistent across them or not. But that's not a very principled way of doing things. And so I don't know if you have better ideas, essentially.
No, I mean, I don't know if okay, maybe less principled. I mean, I like it, but that's actually what we use for other types of things, like the generation of T cells, for example, or the generation of diversity from embryogenesis in hematopoietic stem cells. How do stem cell populations seed, essentially? But I don't know if I'd say, yeah, I mean, we use something similar because it's a hard problem, and I don't think there's any one solution. So, it's an interesting and complex issue that has implications in multiple disciplines.
Yeah, and one thing, yeah, that's right. And again, it feels to me like, that some statisticians must have thought about that, come up with some principles, but I haven't really been able to find something that worked for us.
Right.
One thing that is particular as well about these models is that there is, one part of history, like the recent past, where things are quite informative, right? And we can test a lot of models. And then when we go in the distant past, I mean, essentially we're, I don't know, how do you say, we're running on fumes, in the sense that like there's few enough lineages that are left from the present day diversity that, we're trying to learn from less information. And so there's part, I mean, I guess people have looked at sloppy models that have like stiff and sloppy directions. And maybe it's not that unusual in biology, but for us, it has been challenging to try to, kind of jointly interpret these stiff directions and these sloppy directions, and then translate that to like language of anthropology, for example.
Right. Yeah. Yeah. Perfect. Well, maybe we have time for one more question. If there's anybody who wants to ask it, you can ask it in the chat, or you can even unmute yourself. I'm not sure if I see. Well, I would just like to say thank you again, Simone, because I think this is fascinating, and it obviously has huge implications on our knowledge of ourselves, but I think it has big implications, like I said, for other genetic diversity or other studies of genetic diversity in cells, even though there you might not have diploid populations, but you can understand the evolution throughout it. So thank you so much for sharing this work with us, and we look forward to seeing everyone next month. And actually it's the same date, because March and February always have the same date. So it'll be March 2nd on the Wednesday. So thanks very much, Simone.
Great, thanks for the invitation.
Bye, everyone.
Bye, everyone.